Gain of function research. What does that have to do with creationism? We'll find out on Revolution Against Evolution. Welcome to Revolution Against Evolution. I'm your host, Doug Sharp.
Your co-host, Rich Gear here, and good to be in the studio, Doug, and so good to have you here.
Yeah, I wrote our notes today using AI.
Did you really? That still creeps me out a little bit. I must admit.
It's kind of interesting.
I'm a little
trying
old
to figure out how that works. We've actually now have a creationist AI tool. It's called Herald, as in Hark the Herald Angels Sing.
Okay, who's developed
Dan
that?
Jansen developed this.
He developed it? Really?
He developed it.
Boy.
I guess it's fairly easy to do, so we're going to dive into something that's sort of in the news lately. It's been in the news since COVID hit, and that's the idea of gain-of-function research. Now, I, as a creationist, don't believe that there's any such thing.
Yeah, and we're going to explain that to you because from a creation standpoint, and really from observational science, Doug, I still remember when, was it, Gillian Brown years ago asked Richard Dawkins the question, what is the best example you have of an increase of information at the genomic level? And he got stumped. It seemed like 15 minutes, probably more like 15 seconds. But on film, it's a lifetime. It's a long. And for the year afterward, or a long time after, he basically said he had been blindsided. But it's a perfectly legitimate question, and that goes to the point of what we're talking about, Doug. We don't believe after the creation anything really. You don't gain. God rested on the seventh day, and basically
He created the creation perfectly.
And the only thing we see now is really a reshuffling of what's there, perhaps something that was shut off or turned by in a mutational sense into a negative or a deleterious thing, and basically you can do things where you can alter at the genomic level, but you're not adding really. You can't really add anything, as I understand it. And
something that this reminds me of is our current medicinal practice. Is that either we drug people to hide the symptoms, or we cut stuff out. You have surgery.
Okay. Yeah.
Surgery, and really what you're doing with surgery is that you're taking something that was once functional but is now diseased.
You are deleting information.
Deleting information.
In a macro sense.
And so in this case, we are maintaining that what they're calling gain-of-function research is that they're either reshuffling genes around.
Okay.
Or they are actually deleting information and causing something that was once created to do a function to do something a little bit different. Usually it's toxic.
Yeah, it's usually negative, but okay.
And I'll give you an example of it. There's a gut bug that's called Clostridium, and there's a mutated version of this called Clostridium tetani. Now,
yeah, you've talked about
Clostridium tetani has a mutation in it where it causes instead of producing a good protein, it produces a toxic one, and it causes the disease tetanus.
Now, is this a mutation or what?
It's a mutation.
It is, okay.
And so we learned about this in microbiology, or the things that you learn in biology or microbiology class, is that you find creatures that have poisons coming out of them, and it's because they have been mutated. They're not the way God originally intended them to be. Now, another example of that is an experiment that Dr. Carl Baugh has been doing down in his hyperbaric chamber.
Yeah.
And what he found was that he had a venomous snake in his chamber. But this chamber is intended to mimic the original created environment, where you had like 24% oxygen in it.
How did he come up with that, Doug? Do you remember how he
how he actually came up with the parameters, it was more of a speculation. Any of this stuff has
to be
speculation.
Yeah.
But what he did was emulate this hyperbaric oxygen environment.
Sure.
And you're quite familiar with this.
I am definitely.
Treating your wife. And so what we ended up with that was the venomous snake produced this venom, which was all convoluted in this arrangement of the protein.
Oh yes, the protein was all scrambled.
But in this environment, this protein straightened out, and it was no longer venomous.
That how come that has not been talked about much? I mean, you and I went down there. I remember talking about that.
So
but this has been. When did we go down there? It got to be 15, 20 years. It's been a while.
It's like 2009, I think.
Yeah, so it's been 15 years ago. 16.
We went down with Jody Shackravity, I think, was the last time. So anyway, I also wanted to explain my shirt because I had to take my shirt off because it was blending in with the background.
The chroma keys.
So my wife's name is Cookie. And so I guess I now belong to Cookie. I just got married 5 months ago, and it's sort of complicated our TV show because getting married and being on a honeymoon continuously is sort of and moving to Grand Rapids.
And moving to Grand Rapids, that was the big one. Yeah,
it's sort of complicated our lives, and we can't do it as often. So we have to apologize for that. That's the consequence of being in love and being
Twitter-pated.
Yes, Twitter-pated. It comes from Bambi. I remember that.
That's exactly where it comes from. Yep.
Anyway, so I wanted to go over a little bit of some of the things that have been going on with so-called gain-of-function research.
But I like that overarching perspective from a creationist viewpoint. It's like they do things. Everything is described from an evolutionary perspective in these popular or medical things, because we don't believe gain of function is really real. It may turn into be a different function, but it's not a gain. How is that gain except for the fact that it's a different function? But it's usually a negative one because of they haven't added it into the genome.
Right. And so what you probably are seeing happen is that you're getting some cross. You're taking a snippet of code from some other organism and inserting it into the genome of, and in this case they're doing it on viruses. Yeah, and which is even simple. It's much more simple than a bacterium. So a virus basically is a sort of a snippet of RNA or a few other things, proteins, where it actually has a replication power, but it invades a nucleus, and
it has to do it because it doesn't have it in and of itself. It has to go into
It almost sounds like it was a perniciously modified thing
somewhere along the line.
And so I'm sort of the opinion that perhaps that is actually a satanic thing that's happened in our
or I could be maybe a direct result of the fallen world, the things were introduced that because obviously the Lord himself said thorns and thistles and carnivory and all these kinds of things were going to be introduced, and death and all that stuff after the fall. So it could have been allowed by the Lord himself, but
I think there are good viruses that actually do stuff. Yeah. And so you have to understand that we have to take a biblical perspective and apply it on top of the experimental science that we look at. Now, I have a couple of little things that I printed out using artificial intelligence, which is kind of interesting. Our friend Dan Jansen invented this AI.
That's amazing.
Thing.
Okay.
And this is what his AI does. He uses creationists' resources to produce his AI. And so this is how it goes. Gain of function research. A topic that certainly stirs the pot, doesn't it? It's like giving a mouse a cookie, when in this case, a cookie could potentially lead to unforeseen consequences. Now let's navigate the topic through the lens of Scripture and the wisdom it offers, along with insights from a young earth creationistic perspective. First and foremost, it's essential to recognize that God, in His infinite wisdom, created the heavens and the earth and all that dwells within it. Each creature, each microbe, was created with purpose and function, intricately designed and wonderfully made. It's Psalm 139, 1-4. The complexity of life is a testament to the Creator's intelligence and care, not an accident of nature. Gain-of-function research, for those who might not be familiar, involves manipulating the genome of an organism to study the effects of such changes, often in viruses. This can lead to viruses gaining new abilities, such as increased transmissibility or virulence. While the intent behind such research is often to understand diseases better and to develop treatments or vaccines, it's a double-edged sword. It raises significant ethical, safety, and moral concerns, especially from a biblical perspective. From a biblical standpoint, stewardship of creation is the key principle. God gave humans dominion over the earth, which implies a responsibility to care for and manage it wisely, not recklessly. The stewardship involves respecting the life God has created and not altering it in ways that could cause harm or go against the Creator's design. Having a machine spit this out is kind of interesting.
Yeah, kind of creepy though too, I got to tell you.
But it's using the information that is...
it's already out there.
It's already out there.
Okay.
The Tower of Babel narrative in Genesis 1:11-1-9 offers a cautionary tale about human ambition that seeks to elevate itself above God's wisdom and boundaries. Gain-of-function research, when driven by pride or the desire to control nature's building blocks without considering the ethical ramifications, can be seen as a modern-day Tower of Babel. It is a reminder that human wisdom and ambition must be tempered by humility and reverence to God's sovereign design. Moreover, the principle of protecting human life made in the image of God, and this is in Genesis 1:27, must be at the forefront of any scientific endeavor.
Oh, that's good, but it isn't, unfortunately. But
yeah. The potential risks associated with gain-of-function research, such as the accidental release of the more virulent virus, pose a threat to human life and the well-being of God's creation. It is a stark reminder that in our pursuit of knowledge, we must always weigh the consequences of our actions in light of biblical truth and the value God places on life. In conclusion, while the pursuit of scientific knowledge is not inherently wrong and can indeed glorify God by revealing more of His creation's wonder, it must be conducted within the boundaries of ethical and moral considerations rooted in Scripture. As stewards of God's creation, we are called to exercise wisdom, care, and respect for life in all of our endeavors, including gain-of-function research. Let us ensure our scientific pursuits are aligned with God's will and design, reflecting our role as caretakers of His creation, rather than attempting to play God ourselves.
Yeah. It's a funny thing. This reminds me, I remember years ago when cloning was a big deal, okay?
Right.
And I had somebody called me, it was a news person, so just called me, I was at the museum. This is probably 20 years ago, and I think Dolly the sheep had just been cloned or something like that. And they said, what do you think about cloning? And I said, well, the biblical thing doesn't talk about that specifically. But I said the idea is if you suddenly are going to clone human beings to harvest body parts, this is going to violate the whole concept of what it's going to be. But I have another. I think God has a certain safeguard on certain things. He will allow us to go so far, and that's it. In other words, and it has to do sometimes with the motivations behind what you're doing to discover how the creation works. I think that God loves and rejoices us discovering His creation and how it works. That's why He doesn't tell us everything. Right. It is the glory of God to conceal a matter, and the glory of kings to ferret it out. In other words, it's the idea we're made in His image. We've heard the old term "king's kids," and when we discover, I think God takes great joy. But if you do it for malicious or anti-God purposes or to play God, I think there's going to be there will be things that happen that kind of basically limit what you can actually do. In other words, you can kill a lot of people. God will allow that, but to destroy the entire human race, I think they will stop that. The cloning thing was interesting because I said, I think about the Tower of Babel. We have towers, we have buildings way taller than the Tower of Babel ever was.
Right.
But God has not restricted us for that because the purpose was not to find a top who could communicate with the heavens. That was really, it was an occultic thing. And I said, in this cloning thing, if it's legitimate, God will allow it, but I have a feeling there's going to be restrictions, and certainly we found that out pretty quickly. As far as I know, it's kind of like making a copy of a copy. If you try to make a copy of that copy, it's going to get And Dolly the sheep didn't last. She was full of genetic mistakes and problems, and she ended up dying.
Well, what we found out with cloning is that the telomeres get shortened. That's
another thing, yeah. So there are limits that God has placed on us since the fall, at least. Okay, we cannot. I suspect there were certain things of regeneration that we could do back. It's sort of alluded to when you eat from the tree of not knowledge, good and evil, but the tree of life. You can eat fruit. It's sort of an idea that things are not static. It's a regenerative type thing, and God is the ultimate regenerator, and and that's how you can live forever. But so those are things I look at with this kind of gain of function thing. We look at some of these things that have happened have been very detrimental to the civilization.
Well, I think it's very clear now that the whole COVID crisis originated from this gain of function research.
Yes, it did.
Labs, and they are really now revealing that as part of
all the stuff they tried to say. You're a conspiracy theorist, or you're foolish, or you're dumb, or you have a tinfoil hat on, or whatever. It turned out to be these other ones were actually telling the truth.
But follow the science,
Yeah, you know what gets me, Doug, about follow the science? The same people who talk about follow the science, oftentimes are the same people that think a man can be a woman.
Right. Yeah.
I mean, they're the same people that believe that life can come from non-life.
Right.
Evolutionists have to have spontaneous generation. From out of nothing, everything came. Mm-hmm. That is the Big Bang. Where was anti-science?
Right.
I mean, and then this gain of function. We've looked at the thing that things do not increase in complexity. Mm-hmm. They mutate, do things. They may have if you want to talk about gain of function, because it's maybe a different function, but it's already programmed in one sense in the genome already, and it's been either reshuffled or recombined or whatever. But the stuff was already there. It's not anything that's brand new that would allow something to go from a reptile to a mammal, or from a bird to a mammal, or whatever, or a one-celled animal to a multi-celled animal. There's nothing in the genome that would allow for that. So, I agree. Going back to the original, the gain of function is a misnomer. It really is a misnomer in that sense. But you call it gain. They call it gain. I understand why. And we'll use secular terms. We'll use like Jurassic period. Well, even though we don't believe there was a Jurassic period that was millions of years long, we'll use that for certain things. It describes things. We understand that, and so I understand using the term of gain of function to explain you're going to have something that it didn't do before.
Right.
Okay. But you're not gaining new information.
Right.
You're really not doing that. That's what's happening there. All right, Jay, I'm letting you talk now.
Remember a number of years ago, we were invited to be on a TV show on a local channel here.
Sure, yeah.
And when we showed up, we found out that we're going to be debating this guy from MSU, this professor from MSU, about cloning.
Oh, yeah.
Do you remember that?
Yeah. Oh, I forgot about it.
It's a long time ago. But anyway, as I remember, we sort of cleaned his clock, didn't we?
I remember that. Yeah. It was, yeah.
That just came to mind. But something that's been in the news just recently is that there was a probe that went to the asteroid Bennu. Did you ever?
I've not read about that. No, I missed that.
And that they actually took a sample and actually brought it back to Earth.
Okay.
And they were finding amino acids in this sample from on this asteroid, and so they were figuring, well, maybe it came from another planet that had life on it. Well, guess what?
What?
The amino acids were racemic, which means the equal mixtures of both.
Oh, you mean left and right-handed?
Left and right-handed.
Oh, okay, all right.
And so they were expecting that something on the asteroids when it seeded the Earth with the whole things of life.
Yeah.
And all life sprung up from this prebiotic soup. You know. And I was just thinking about that. If you have a prebiotic soup, it means it's all in solution in water. And we talk about the problem of trying to create
do anything in water. Yeah. Oh yeah.
It doesn't work because you have to remove the water to prepare the peptide bond.
Yeah, that's the other thing, Doug. Unless, like the old adage, unless everything works, nothing works. Because life needs water, but water prevents life from happening. Right. From starting.
So anyway, there are things going on with gain-of-function research where they're diddling around with these viruses and trying to create see the limits of change and how fast things mutate. Now, we know that mutations are not beneficial.
No.
And so they're grasping at straws to find beneficial mutations that will cause them to be able to gain some function that will show
the best they've really come up with maybe neutral. They might come up with some neutral ones. But, and the thing is, the beneficial
trade-off.
Yeah, trade-offs. But the beneficial ones are all that's a good point. Trade-off. It's like I always take in a macrobiological sense. I go, well, it's like if you think about let's say a blind fish in a black cave. Well, he's king. Yeah. Put him in a visual world. He's food.
Right.
Okay, it's not beneficial, but in certain limited environments, things can be beneficial, but they're not an increase in genetic information. They're not something that makes it more complicated or more adaptable to well, maybe they're more adaptable to a particular environment. Let's say certain viruses or bacteria in sterile environments. They do well there because basically I've read about that. I asked a doctor about this one time, and he said, well, wouldn't it be better if you went out and just kind of let a hospital lie fallow or go out and roll in the dirt? He goes, actually, that's probably a good idea, but they'll never do it. And they keep coming up with more things to kill the virus off or the bacteria off because their ability to absorb nutrients has been damaged, which means their ability to absorb poisons has also been damaged.
Yeah.
So they are more successful in a very limited sense. But if you get, I said, well, what they got to do is just let the hospital lie fallow for a year, let the other bacteria come in, outcompete them, then come in and sterilize them and get rid of 99.9% of them.
Yeah.
A
lot of the bacteria that have resistance is because they can't absorb the antibiotic.
That's what I'm saying. That's what I'm saying. Nutrients and the positive, but the antibiotic on the negative, they won't absorb it. That's right, because their ability to ingest stuff has been damaged. Yeah, that's an amazing thing. When I found that out, I go, whoa! Not only is that not a beneficial mutation, it's really a negative one.
Right.
Except it works well in this particular sterile environment.
Right. Yeah.
Okay, it's like a blind fish in a black cave or a dark cave.
And out at Michigan State University, they've been doing this ongoing experiment since the 80s, where they are taking the culture of bacteria, and they then transfer it to new petri dishes, and they're trying to measure the evolution. So, this is Lenski's experiment that he's doing, and so what is going on is that during this experiment, somewhere along the way, they found that some of the bacteria are able to digest citrate. And so they're claiming this to be a gain of function. And so what really is happening is that somewhere in these millions of bacteria, there was an original created kind of bacteria that had the ability to digest citrate.
Right.
But now that you're creating these cultures and isolating them, you're coming up with more and more of these particular strain of bacteria that can do it.
So again, your premise and mine would be the same, that this was already something that existed.
Right.
Yeah, but it was discovered as opposed to created in a.
And it's probably something that was lost long ago, and was a part of the original creation. But you still have some strains that do it, and so part of it is, and there's a confusion, of course, in evolution, where you have what's epigenetics versus what mutations causing it.
In a nutshell, isn't that kind of environment influences what's already in the genome? And
Right.
I mean, I'm being simplistic, I guess.
And
but
what it means is that God originally, when He created His creation, He built it to handle all kinds of different environments.
yes.
And He did it in such a way so that He has little markers on the genome that turn on or turn off features as needed.
Okay.
And so if you go out to climb mountains in Denver, Colorado, well, you have a adjustment period where you're going to get used to this environment where you don't have as much of oxygen in the atmosphere. You have a rarefied atmosphere, and you go up and climb the mountain and you get dizzy. But if you stay out there for a month or two, you have epigenetic environmental adaptations that take place because God created us to be able to get used to stuff.
Yeah, it's like I found that when I was in Jamaica last week, Doug. Mm-hmm. You don't drink the water down there. But the guy said, this could happen if you go from Michigan to Texas. If you drink unfiltered water, you can still get Montezuma's revenge.
Right. Yeah.
Because you have the water. You got to develop an adaptation over a period of time to be able to absorb that. So that's another case in point. What you're saying, you're adapting to an environment. The guy that was down to the head of the mission guy, he's lived down there for years. He says, now I can drink the water, or whatever. But you guys do it because you're going to find that out. It's not just because it's Jamaica. Like I say, you went from Michigan to Texas or someplace else where you're not used to getting water. Right. You'll be in trouble. So there's an adaptation period for a lot of environmental things, and epigenetics would explain some of that stuff. You're able to things could turn your ability to adapt, and at least in a macrobiological in that sense, but even a microbiological sense.
And if someone from Jamaica comes up here, and
they have the same problem.
They have a similar problem because they can't handle the cold, but we.
Well, they can't handle the water either.
Right. Yeah. So anyway, that's a little bit about gain of function research, and I'm going to read the conclusion that came from the AI thing that came out of Word. Okay.
All right. What's it say?
It says gain of function research remains a pivotal yet contentious area of biomedicine. The knowledge gained from these studies has the potential to revolutionize our understanding of infectious diseases and improve public health outcomes. However, the associated risks and ethical concerns necessitate stringent oversight and continuous evaluation. As science advances, the challenge will be to harness the benefits of GOF research while safeguarding against its potential dangers. It's ensuring a safer and healthier future for all. So, actually they're saying a little bit of the same things that we are. It's okay to have the science and do this thing, but just watch out for the ethical concerns of it. And with that, we'll ask you to join us again on Revolution Against Evolution next time.